Okay, so welcome to this next video in which we are discussing the lectin complement pathway. So we've discussed that when a microbe is invading a portion of tissue, what that will lead to is the inflammatory response. Okay, and the inflammatory response leads to type 1 and type 2 activation of the endothelial cells of the capillaries and venules, which supply this affected portion of tissue. And those endothelial cells in the capillaries and venules will then open up gaps in between neighbouring endothelial cells, and this will allow fluid from the blood and also proteins to move into the interstitial fluid. Now these holes aren't generally big enough for leukocytes or cells to move through, okay, at least not without help, but it will allow proteins to move into the interstitial fluid, and this is important because it's going to allow complement proteins to move in. Okay, right, so let's discuss now how we initiate the lectin complement pathway. So, we need to discuss a protein complex known as the mannose-binding lectin complex. Okay, so this is the star of the show, but this is not a complement protein, so this is another protein complex where the components are produced by the liver, but they are not complement proteins. So the mannose-binding lectin. And we're going to discuss the mannose-binding lectin complex. Now there will be a protein that is actually known as the mannose-binding lectin. However, it's going to be the mannose-binding lectin complex which is important, so it's the whole complex that we're going to look at. So, let's have a look at the structure of this complex. So this complex has a structure that we will try to draw. So, how should I draw this? I'll start with it like here. So you have these. Well, actually, I'll start with what mannose-binding lectin is. So you have many proteins called mannose-binding lectin proteins which are going to assemble together to make this complex. So you usually assemble around 6 mannose-binding lectin proteins to actually make the mannose-binding lectin complex. Okay, so this here is a single mannose-binding lectin. Ignore this other line, I was about to draw another one, but this single structure where you've got this tail and a head, this is a single mannose-binding lectin. And mannose-binding lectin is often abbreviated to MBL for short. And these molecules are created within the liver. Okay, right, so we're then going to bind 5 more of these things together to make a nice complex. Okay, so there's one, and I'll put another one here. Okay, another one here, and then finally, well, sorry, not finally, penultimately, another one there, and another one here. Now we're finally done. Okay, so we have these 6 mannose-binding lectin molecules complexed together to make our mannose-binding lectin complex. However, that's not the mannose-binding lectin complex complete. You also have 4 other proteins bound. Okay, so let me show these. So at the bottom here, we'll put 4 more proteins. Now, basically, even though we've got 4 proteins, there's only 2 separate types here. So, let me colour these in a little bit. So, 2 of them are basically of the same type, and then the other 2 are also of the same type. So these 2 that I'm colouring in green, they are what are known as MASP2 proteins. Okay, so their name is MASP2. Now, what does MASP2 stand for? Well, it stands for mannose-binding lectin. Okay, so that's the M in their name, mannose-binding lectin, and then associated serine protease. So mannose-binding lectin, and then associated serine protease, and this is specifically the mannose-binding lectin associated serine protease 2. Okay, so we're also going to see MASP1, which will be the mannose-binding lectin associated serine protease 1. Okay, so you have 2 MASP2 proteins bound to the base of these 6 mannose-binding lectin proteins, which are all complex together. Okay, and then below that, which I'll colour in vivid purple here, these are MASP1 proteins. So you have two MASP1 proteins here as well. So two mannose-binding lectin associated serine protease 1 proteins. Okay, and this entire thing now is the mannose-binding lectin complex, or the MBL complex for short. I should also add that you will occasionally hear people refer to this as the mannan-binding lectin. Instead of mannose, occasionally people will write mannan. Okay, so I will explain to you what mannose is. Mannose is a monosaccharide, the structure of which I will show you in a moment. But mannan is the polysaccharide where you polymerize mannose with mannose with mannose with mannose with mannose with mannose. So you stick loads and loads of mannoses together to get mannan. Okay, so mannose is the monosaccharide, mannan is the polysaccharide. These proteins actually really bind to mannose. They will bind to a mannan because mannans have mannose in, but it's better to call them mannose-binding lectin. But you will hear people refer to it as the mannan-binding lectin. Right, okay, so what does this protein complex do? Well, basically, this protein complex is within the blood. All of these proteins are assembled together in the blood. And now, when we increase the leakiness of our capillaries and our venules that are supplying this infected area, then what's going to happen is this mannose-binding lectin complex is going to come out of the blood and it's going to meet the microbes. Now, a whole bunch of microbes have structures on their surface to which this mannose-binding lectin complex can bind. And just to give you a flavour of the the variety of microbes which this complex will be able to bind to, you have bacteria, of which the main examples which this is very effective against would be Salmonella. Okay, so a famous gastroenterological bacterium which causes food poisoning often. Listeria, another gastroenteritis causing bacteria. Then you also have Neisseria species, of which the two main ones would be Neisseria meningitidis, which causes meningitis in children generally, and Neisseria gonorrhoeae, which causes gonorrhea. Okay, but other than just bacteria, you also have fungal infections. So, for instance, Candida albicans will have a target for this mannose-binding lectin complex. Candida, we all have it in our mouths generally, but if you're immunosuppressed, if you've got HIV, for instance, then what can happen is it can completely overgrow and you effectively get a huge amount of this fungus growing in the back of your throat and also in your esophagus, okay, which is very dangerous. Then it can also bind to the fungus Cryptococcus neoformans, which is quite a rare cause of pneumonia. It causes meningitis. It causes cryptococcal meningitis. Okay, and also it can actually bind to viruses. Certain viruses will also have targets for this mannose-binding lectin. For instance, human immunodeficiency virus 1 has targets for the mannose-binding lectin complex. In addition, respiratory syncytial virus, RSV, also has targets for this. So the point of that discussion was just to say that many different forms of microbe will have targets which mannose-binding lectin complex can bind to. So we've got bacteria, fungi, and viruses. So that's why I'm keeping it nice and general with just putting a microbe there and not saying specifically what sort of a microbe it is. Okay, right. So what does the mannose-binding lectin complex actually bind to on the surface of these microbes? Well, it binds to glycoproteins and carbohydrates which have either mannose or glucose monosaccharides at the end of the carbohydrates. So let me show you the structure of glucose and mannose so that you can understand what the difference between the two are. Okay, because if you're not armed for this, you will think that mannose and glucose are exactly the same thing, but they're not. They are different optical isomers of the same molecule, if you like, but they are different molecules. They have the same elements making them up, but they have a different optical isomerism. Okay, so let me show you the structure of glucose firstly. So we'll start off with glucose. Okay, right, so glucose is this six-membered ring, and we'll draw skeletal structures. So remember, you don't show carbons in skeletal structures, you show them as corners, and you don't show hydrogens coming off carbons. Okay, so here is this six-membered ring here, and one of the members is an oxygen, and the 5 other members are carbons, which means we don't show them, they're implicit. Okay, then the 6th carbon comes off up here. Now, what we're going to have to do is show the direction in which it's coming off. So imagine that this ring is in the plane of the piece of paper, and then this 6th carbon is coming out towards us, and the way that you show it coming out towards us is that you put this expanding bond, basically. It gets thicker as you go along, and that denotes that it's coming out of the page at us. Now, I'll draw this, I'll sort of I'll overlook the fact that we're drawing a skeletal structure temporarily here, because it's easier on the eye for this bit just to not use skeletal formulae. So I'm just going to put CH2OH here, because it just looks a little bit odd if you then just put an alcohol group sticking off. It looks scarier if you put the alcohol group coming off than it does if you just put CH2OH there. Okay, skeletal structures, we use them because they actually make things easier on the eye. So when they don't make things easier on the eye, we can bend the rules a little bit. Okay, so in this case, it looks a little bit scarier not to just put CH2OH there, and instead just to obey the strict skeletal formulae. Okay, so we're sort of doing a half skeletal, half molecular formulae drawing here. Right, okay, so the methylene group with the alcohol group comes off, coming out of the page towards us. And of course this hydrogen here, sorry, this carbon here, it's only got 3 bonds so far. So because we're drawing in a skeletal structure, the final bond is implicitly to a hydrogen atom, okay? So we don't show that, but the hydrogen atom will be going off into the board. If this group's coming out of the page towards us, then the other bond must be going into the page away from us. So the hydrogen will be going into the board, into the page away from us. Sorry about saying it's a board, it's a page. Right, okay, so now let's continue on. So down here, we then have an alcohol group which goes into the page away from us. Okay, so off this carbon here, which is the 4th carbon of our glucose rings, you start off with this one, 1, 2, 3, 4, 5, 6. Okay, this has an alcohol group that goes into the page away from us. Therefore, the hydrogen that must also come off this carbon is coming out of the page towards us. Okay, then we move down here. This 3rd carbon also has an alcohol group coming out of the page towards us. Okay, and you might wonder, well, why am I stressing this so much? Because this is how glucose and mannose are going to be different from one another. The way in which these alcohol groups are arranged, i.e. whether they're coming into or out of the board, is what's going to be different in mannose, I'm afraid. That's all that's going to be different in mannose. And if you don't understand it, then you won't understand how mannose is a separate molecule from glucose. Okay, so off this 3rd carbon you have an alcohol group and also a hydrogen coming off it. The alcohol group comes out of the page at us, and the hydrogen goes into the page away from us. Okay, then off this 2nd carbon the same thing occurs. You've got an alcohol group and a hydrogen. The alcohol group goes into the page away from us, the hydrogen comes out of the page at us. Now, there are 2 separate molecules that are both referred to as glucose, okay? And they differ in whether the alcohol group of this 1st carbon comes out of the page at us or into the page away from us. So if you put the alcohol group here going into the page away from us, and the hydrogen coming out of the page at us therefore, then that's what's known as alpha glucose. If, however, you have the alcohol group coming out of the page at us, okay, like so, and the hydrogen atom therefore going into the page away from us, that's what's known as beta glucose. So there are two separate molecules of glucose, alpha glucose and beta glucose. Now, because there are two separate ways this can be, on the picture people will often put this odd sort of curvy thing like that, and then they'll put the alcohol group there, and they wouldn't imagine that bit there isn't there now, just that bit that let me cover this up with another piece of paper so that you can see. So imagine that bit's gone. If you just draw that, that's what people will generally draw for glucose. They draw this squiggly line here to denote that it's either going into the page away from us, in which case it's the alpha isomer, or it's coming out of the page at us, and in which case it's the beta isomer. So that's all that denotes. Right, so that's glucose. Now let's discuss mannose. Okay, so it's an almost identical structure. All it's going to differ by is one bond, in fact. Okay, so mannose, again, it's this six-membered ring. So here we go, where one of the members is oxygen and the other 5 members are carbons. Okay, here we go, and then the 6th carbon comes off up here, and again it's coming out of the page at us. Okay, so this is exactly the same as before, CH2OH up there. Then off this 4th carbon, the alcohol group goes into the page away from us. Okay, let me just add a bit in there. Off this 3rd carbon down here, the alcohol group comes out of the page at us, and the hydrogen therefore goes into the page away from us. And then it's the alcohol group off this 2nd carbon here that's different. It's now going to come out of the page at us. That is all that is different between mannose and glucose. It's this alcohol group off the 2nd carbon. It now comes out of the page at us. And these are different molecules. You cannot change whether the alcohol group is going into the page and the hydrogen is coming out of the page for this scenario where the alcohol group is coming out of the page and the hydrogen going into the page, not without doing a chemical reaction. So these structures are rigid, basically. You can't just swap those two around, not without breaking both bonds and physically swapping them. Okay, and again, the alcohol group can be in either position. Okay, so if the alcohol group goes into the page away from us, that's what's known as alpha mannose, just like alpha glucose. So you keep the exact same naming system. And if the alcohol group comes out of the page towards us, that's what's known as beta mannose. So you have two isomers even of mannose. Right, okay, so that's the difference between glucose and mannose. It's just this alcohol group on the 2nd carbon of that ring. In glucose it goes into the page away from us, in mannose it comes out of the page at us. Right, okay, so it is these monosaccharides here, and I'll put that word down because it's a very nice keyword. Okay, if they still made keyword sheets at A-level, at degree level, this would be one of the words on the keyword sheet. Right, so monosaccharide just means that it's a monomer of a polysaccharide, basically. So it's the subunit that makes up carbohydrates, which are polysaccharides. So, basically, you can have these structures within carbohydrates. You can also have them on glycoproteins. Let me just explain this. So, in carbohydrate molecules, what you do is you take these monomers. Okay, so I'm just going to now draw one of these monosaccharides just as a single box, and you can join them together, one after the other, and create what's known as a polysaccharide, or what's also called a carbohydrate. Carbohydrate is the obviously the more pervasive name for a polysaccharide. It's the one that the general public uses, okay, knows and yet loves. Right, so sugars is maybe even a more common name for this. So sugar means carbohydrate. Obviously, I'm not talking about the white stuff, the white crystal that people think of when they're talking about table sugar. But when we're referring to sugars in general, what this means is, what am I writing here? That's useless. Carbohydrates. Okay, and these basically are these polysaccharides. They're polymers of these monosaccharides. Okay, so sugar, carbohydrates, or maybe you'd prefer to use the word polysaccharides. Okay, so you polymerize these monosaccharides together, and you're not just polymerizing glucose and mannose together, you've got a whole plethora of sugar molecules that you can use. However, the important thing to understand is that carbohydrate molecules are put on the surface of microbes. So if we've got our microbe here, maybe it's a fungus, maybe it's a bacterium, maybe it's a virus. Whatever it is, often they have carbohydrates on their surfaces. So let's have this carbohydrate shown by this green line. So this represents a polysaccharide. Now, right at the end of these polysaccharides, so let's say this is the end of the polysaccharide, if you have a mannose or a glucose monosaccharide right at the end of the polysaccharide, then that is what the mannose-binding lectin complex is going to bind to. Alternatively, you can have what are known as glycoproteins on the surface of the microbe. Okay, so what's a glycoprotein? Well, basically, this is where you put a protein on the surface of the microbe, and then to the protein you attach little polysaccharides, often sometimes known as oligosaccharides. So generally polysaccharides implies that you've got hundreds of these monosaccharides attached together, basically. So I was too lazy to draw hundreds of the things. I just drew 6. This would not be counted as a polysaccharide. A polysaccharide, you need hundreds of the things. However, when you just have a short length, like what I've drawn, this might be referred to as an oligosaccharide. Okay, so short little polymers of monosaccharides, such as the one I've drawn above, where you've got 6 bound together, they're often referred to as oligosaccharides. Okay, and you can attach oligosaccharides to proteins and have them sort of dangling off here. And when you attach an oligosaccharide to a protein, that protein is now referred to as a glycoprotein. Okay, because it's got a sugar molecule. That's what glyco means, sugar, protein. Okay, so it's a protein that's had a sugar molecule attached to it. So the entire thing now is referred to as a glycoprotein. Let me highlight this up. Okay, so this is a glycoprotein, and I'm going to circle the entire glycoprotein in blue. Okay, right. Now, these oligosaccharides that are dangling off the edge of the glycoprotein, they, just like the polysaccharides, will have terminal monosaccharides. It has to end at some point. So there will be a sugar molecule, a monosaccharide, that is right on the end of the oligosaccharide. Okay? Now, if that is mannose or glucose on the end there, then mannose-binding lectin complex can bind to that mannose or glucose. So they bind to terminal mannose and glucose monosaccharides in carbohydrates and glycoproteins that are on the surface of the microbe. Okay, so we'll continue this discussion in the next video.